All right, Chapter 8. This is skeletal muscle, the structure and function of said muscles. So we will get right into it. And first off, I want to talk a little bit about some of the structure of the muscle. If you look at the entire muscle itself, and we'll see a figure here in a second. If you look at the entire muscle, it's surrounded by basically a thin layer of connective tissue known as the epimysium. And it's going to consist of a bunch of bundles of fibers, basically, of muscle tissue, known as fasciculi. These fasciculi are then surrounded by another layer of tissue known as the perimysium. Basically, these are just layers of connective tissue, muscle tissue, connective tissue, muscle tissue. And then that's when we get into the fibers themselves. Now the muscle fibers then are going to be surrounded by the endomysium. And these are going to be consistent of myofibrils. And they are going to be divided into what's known as sarcomeres. And this is where we're actually going to get into the muscular contraction. This is where the heart of everything is going to come from. Once the sarcomeres, and we'll see here later on, the sarcomeres are going to shorten. And then that's what's going to shorten everything else. So the figure from your book, and if you look at any anatomy, physiology textbook, ex-phys book, you're going to see a figure similar to this, if not the same one. So the muscles being connected to the bones by a tendon, and surrounding the entire muscle, you have the endomysium. And then you can see the various fasciculi. All right. And in between all of these little fasciculi is going to be the perimysium. Again, just connective tissue, just barriers between each of the fasciculi. Now, as we get down a little bit further into the fasciculi, they're going to be comprised of a bunch of muscle fibers, right, or the myofibrils. And then we get further down, and this is where the good stuff's going to happen. We're going to have the muscular contraction occurring in the myofibril itself with the sarcomeres. And we're going to have a couple different proteins that are basically going to be sliding across each other so we can get some muscular contraction. Now, the plasma lemma is known as, or the plasma lemma is the cell membrane. This is what's going to fuse with the tendon itself. And it within it, it actually is what is going to conduct the action potential. The action potential is basically going to be the electrical current that's flowing down into the muscle fiber. And the electrical current's very important because if we didn't have the electrical current, we wouldn't be able to release a certain element. And we'll see what that element is here in a little while. Also helps maintain pH levels and also the transportation of a lot of the nutrients that are going to be transported within the muscle that we need for muscular contraction and for fuel as well, and to build up tissues. Now, there's also going to be some satellite cells that are kind of floating around the muscle fibers themselves. And the satellite cells are basically like the stem cells for the muscle. Anytime a muscle is going to be damaged, such as when you're in a training regimen, you're going to be breaking down some of that muscular tissue. These satellite cells become activated, and they will help basically build the muscle fibers back up. They'll either give a muscle fiber a new nucleus, so they can build the cell bigger and some of its material, or it can actually branch off and create its own cell. And those are two different types of muscular growth. The first one that I mentioned, growing the muscle fiber, each individual muscle fiber a little bit bigger, is known as hypertrophy. Creating new muscle fibers, right, more and more of them, is known as hyperplasia. Most often in humans, it's going to be hypertrophy. Hyperplasia might occur to a certain extent, but it's going to be fairly insignificant. At least that's what a lot of the research is suggesting. So it may or may not occur all that much in humans. In some other animals, it does occur. Cats, we know it occurs. In birds, I believe. Some birds will grow new muscle fibers. But in humans, generally speaking, we're just going to grow each of the individual muscle fibers. And this is, again, where the satellite cells come into play. Now, the sarcoplasm, this is essentially the cytoplasm of the cell. All right, this is where the glycogen is going to be stored. We talked a lot about glycogen in the past chapter. Also, the myoglobin. The myoglobin is important because this is the oxygen transport cell within the muscle. And it is very similar to hemoglobin. There's a few slight differences between myoglobin and hemoglobin, but essentially they're the same thing. They're going to carry the oxygen throughout the muscle so that the muscles have oxygen to go through oxidative phosphorylation, as well as picking up carbon dioxide and transporting that to the blood. Now, there's also going to be structures known as transverse tubules or T-tubules. These are going to carry action potentials, those electrical currents down deep into the muscle. And why this is important, and what I was alluding to earlier, oh boy, my voice just kind of squeaked there a little bit. Isn't that going to be wonderful? So what I was alluding to earlier was the sarcoplasmic reticulum is going to store calcium. And these calcium ions are going to be released with the stimulation from that action potential. So that's going to be important. And the calcium, as we'll see later, is basically going to start the cascade of muscular contraction. Now, it's not the very first thing that occurs. We need some type of signal coming from the brain and electrical current flowing down, and that's what's going to kind of cause all this. But as soon as the calcium is released, then we're going to get, potentially, as long as everything's working correctly, we're going to get muscular contraction. All right, another figure from your book. And again, if you look in any anatomy physiology textbook, any ex-phys textbook, you're going to see a similar figure, picture, diagram. All right, so we have this little bed. I'm not even sure how to describe it. It almost looks like a web, essentially. This blue web is the sarcoplasmic reticulum, and this is where the calcium is going to be stored. Now, in the middle, basically this blue tube and also purple tube, I guess, that is going to be the transverse tubule. All right. Now, the book breaks it up into a couple different categories, but basically all you need to know is that is the transverse tubule. Now, I'm not going to have you label anything on the exam. What I want you to know is why the T-tubule is important. And why it's important is because the electrical current flowing down the transverse tubule will stimulate the sarcoplasmic reticulum, thus releasing calcium. Right, and that's kind of the main purpose of showing this figure. Now, once we get down into the myofibril itself, you can see it's starting to look a little funky. So you're going to have what's known as a sarcomere, and that's basically from this jagged line or Z line to another Z line. So what's going to happen during muscular contraction is this sarcomere is going to shorten. And what's going to happen is we're going to have two proteins. You can see the thinner, kind of lighter type of protein, maybe like a light blue, and then you also have the red here in the middle. Those two proteins are going to slide over top of each other. So when we have muscular contraction, the Z lines become closer together, is essentially what's happening. Now, the two proteins that are sliding across each other are myosin and actin. All right, myosin is this thick reddish color, and then the lighter blue is going to be the actin, and that's also known as the thin filament. Now, another thing to point out is that in the middle, directly in the middle between each of the Z lines is going to be known as the M line, or the middle line, right? So during muscular contraction, these myosin filaments are going to attach to the actin, and the myosin is going to be pulling the actin towards the middle, or towards the M line, thus creating a shorter sarcomere. And every single one of those sarcomeres within that myofiber are going to contract. They're going to get shorter, thus translating into the whole muscle getting shorter. All right, this is a figure actually from a different textbook, but it's essentially what it was describing on the previous slide there. So we have actin up here. It actually almost looks like DNA. It's like a helix structure. And then we have myosin, and you can see the little appendages on it. Those are known as the myosin heads. All right, and those are depicted up here. Now I can see the M line, and you can see each of the Z discs on here on the right and the left. Now these Z discs are basically holding all this together, each sarcomere together. All right, so these Z discs are going to be important when we're training. So what's going to happen is while we're training, while we're flexing the muscles, all right, and we're training properly, and we're exercising, these Z discs will actually break down. Now, they look like really simple structures, at least in this figure, but what's actually going on here is there's about 45 to maybe 50 types of protein, some connective tissues as well, holding all of this together. And those proteins start to break up a little bit. And when those break up, then we're going to have a response to bring in some more protein to build those tissues up, as well as clear some of that out. So this is where, when I talk about muscular damage while we're exercising, this is what I'm talking about, is these Z discs will actually start to break down a little bit. Now, another thing to keep in mind here from this figure is this little picture up here, is that we have two other proteins that are basically connected to actin, or the thin filament. So we have tropomyosin, which is a long, stringy green, at least in this figure, protein that is surrounding and flowing with the actin. And then we also have, on top of that, troponin, and it's usually represented as kind of a blob on top of tropomyosin. Now, what's going to happen during muscular contraction is that tropomyosin is actually blocked, all right, to begin with, or is blocking the binding site of these myosin heads. These myosin heads are directly underneath the tropomyosin. What's going to happen is the release of calcium will cause a binding of calcium to troponin, and this will actually move out of the way. Tropomyosin then will move out of the way, and we can actually have binding. And we'll see that here in a little bit. All right, so let's take a step back here and look at motor units. So we have motor neurons that innervate the muscle fibers. We have to have some type of electrical current flowing down the T-tubules to stimulate the sarcoplasmic reticulum. All right, so the motor units are essentially, and this is their definition, they are the motor neuron in all the muscle fibers that they innervate. If you have more motor units, you're going to have more contractile force. Some muscle fibers, or some motor units, only have 3, 4, 5 muscle fibers connected to them. Some motor units might have 1 or 2,000. All right, it's just going to depend. The more the motor units, or the more the muscle fibers that are associated with the motor units, the more gross motor movement you're going to have, such as those in the legs. The bigger muscles in the legs are going to have a lot of muscle fibers connected to each motor neuron. The motor units within, like the eye or the fingers, all right, that have fine movements and very intricate movements, those are going to have fewer muscle fibers associated with each motor unit. Excuse me. Now, the junction between that neuron and the muscle fiber is known as the neuromuscular junction. All right, so this is where the communication is actually going to occur. Now, there's going to be a problem here, though. It doesn't directly connect with it. There's going to be a little bit of a gap, and this gap is known as a synapse. So we're going to have to transmit that signal across that gap or that synapse somehow, and we'll see how we do that here in a second. All right, so this is what I was talking about. This whole figure here is one motor unit. So we have the neuron body or the cell body, and we have the neuron coming down and connecting to muscle fibers. So in this case, it'd be 3 muscle fibers. All right, so way down here, this is where the neuromuscular junction is going to be. All right, so here comes the concept of excitation-contraction coupling. Basically, what it is describing is the contraction of the muscle due to electrical stimulation. All right, so the action potential was starting in the brain. We're thinking about moving. All right, we need to move our hand or arm for some reason. That action potential goes from the brain down to the muscles in the arm. And that action potential, that electrical current arrives at the axon terminal, right, which is where that synapse is going to be. All right, so that synapse and the axon terminal are going to coincide with each other. It's going to release a chemical known as acetylcholine, also known as acetylcholine. It's pronounced a couple different ways. But this is very important because this is how we're going to actually get that electrical current across that synapse, so we can actually continue that electrical current down through the T-tubule and stimulate the sarcoplasmic reticulum. However, we can't just jump across, release acetylcholine and then go straight to the T-tubule. It needs to go across that synapse, then it binds to a receptor, and then the electrical current can now continue to flow down through the T-tubules. Then calcium then will be released from the sarcoplasmic reticulum. And then this will enable actin-myosin contraction then. So again, the excitation-contraction coupling is basically just describing the electrical signal flowing down through the motor unit and those cascade of events causing the calcium to be released and enabling the actin and myosin contraction, the muscular contraction of the sarcomere. That is what excitation-contraction coupling is. All right, so this was the synapse that I was describing. So this gap right in here is a synapse. So the electrical current is flowing down through the neuron. It can't just go straight directly into the muscle. All right, so there needs to be a release of acetylcholine. The acetylcholine will then bind to receptors on what's known as the plasma lemma. And then this is going to continue then and basically excite and propagate the signal down through the transverse tubule. All right, so let's look a little bit more at calcium. Because again, this is very important. Once we have the electrical signal going down to the T-tubule, stimulating the sarcoplasmic reticulum, we have this huge release of calcium from the SR. And then the calcium then is going to bind to troponin on the actin. So again, actin is the green spheres, and this kind of pink stringy protein is going to be tropomyosin. And then troponin is going to be the gold oblong-shaped protein-looking things. So the calcium is going to bind to troponin. And the reason this is important, like I was saying before, is that during relaxation, muscular relaxation, the binding site for this myosin head is going to be covered up by troponin, tropomyosin. All right, now this is sometimes referred to as a troponin tropomyosin complex, those two proteins together, because they are kind of really a complex. They act basically together. If calcium binds to troponin, troponin and tropomyosin are going to move out of the way. So once the calcium binds to troponin, it can then move that troponin tropomyosin complex out of the way. Then it reveals the binding site so that the myosin can actually attach. Now, there's a couple other things we need to go over before we get to muscular contraction. But basically, the calcium binding to troponin, moving everything out of the way so that the binding sites are revealed on actin is an important aspect of this. All right, so let's go back to this figure from the beginning of the chapter. So again, our main focus here is we need to shorten this sarcomere. And since the myosin, the red, has these little heads attached to it, they can attach to the actin because the actin has the binding sites on them as well. But again, we need to move troponin, tropomyosin out of the way. But again, just to remind you of what we're trying to do here is just simply shorten that distance between the Z line and the Z line. So again, actin, it also known as thin filament. It has the myosin binding site. It also has troponin and tropomyosin that are covering up the binding site for myosin. And we need to move those out of the way, and calcium is going to do that. Then myosin has those globular heads that will then attach to that binding site. Now, during muscular relaxation, there is no actin-myosin binding. All right, and that's really what relaxation is that actin and myosin are not bound together. Now, when it's contracted, when the muscle is contracted, there's a power stroke, meaning that myosin has pulled actin towards the center or the M line. So everything, the sarcomeres, the myofibrils, the muscle fiber itself are all going to shorten. And that's why if you take your bicep and do a bicep curl, fully contracted up near the top, right, the muscle feels shorter. You can even see it shortening up, right? And when you extend it, it lengthens, all right? And that's basically what's happening. Since all those sarcomeres, all those thousands and thousands of sarcomeres are shortening, the whole muscle itself shortens. Now, after the muscle has been contracted and everything's done and we start to relax, the myosin detaches. It moves back to a cocked position, kind of ready to, attach to a new binding site and perform more muscular contraction if need be. Also, during relaxation, the calcium then is going to be transported back to the sarcoplasmic reticulum and saved for a later time. All right, so energy for contraction, our good buddy ATP, adenosine triphosphate. We've talked about this guy quite a bit, or gal. I'm not really sure what ATP really is, but it's a thing. It's probably not a person, so I probably shouldn't be describing it as such. But anyway, this is going to bind to the myosin head, and this is the whole purpose of why we need ATP in the muscle. All right, while we go through that oxidative phosphorylation so we can continue exercise and continue muscular contraction is because we need to continue to make ATP so we can get the energy out of it so we can actually have muscular contraction. So the ATP binds to the myosin head, and on the myosin head, there's going to be an enzyme that hydrolyzes it, basically breaking up ATP, and it breaks it up into ADP and one inorganic phosphate. Now, again, during muscular relaxation, the X potential, that electrical stimulation is going to end. So that stimulation is going to end in the sarcoplasmic reticulum. Thus, calcium going back to the SR as well. Also, there's going to be some more ATP binding, and we'll see that here in a second. So, an overview of the sliding filament process. Sometimes you'll see it as the sliding filament theory. Sometimes you'll see it as sliding filament process. I'm not sure if it's really a theory anymore. I mean, I think there's a few things that need to be worked out and decided what's actually happening, and maybe a little bit more research, but it's pretty much fact now. This is how we know the muscles contract. All right. So again, we're going to have some type of stimulation from the brain, and it's going to go down through the motor units, down to the neuron, to the synapse. Acetylcholine is going to be released, go across that synapse, bind to a receptor on the plasma. There is going to be electrical current going down through the transverse tubule, releasing calcium from the sarcoplasmic reticulum. That calcium binds to troponin, where there's going to be now a conformational change that occurs where the tropomyosin changes shape and basically revealing the binding site so that myosin can now attach. Now, ATP that's bound to the myosin head is hydrolyzed by ATPase. This energizes the myosin head. It gives it a potential energy to do what it needs to do, basically contract or pull actin towards the middle. So with the release of the inorganic phosphate and the ADP, the myosin head will move towards the M line. So it's not the hydrolyzation or the breaking up of ATP into ADP and PI. It's basically breaking it up and then releasing it is what causes the energy to be released. Now, at the end of that muscular contraction, another ATP is going to bind. All right, so ATP needs to bind so that we can have muscular relaxation. If ATP does not bind, we're going to continuously contract the muscles until an ATP shows up, or until some enzymes start breaking up the bonds between the muscles, which is actually what happens after we die. It's a process known as rigor mortis. When we die, approximately 24 hours or so after we're gone, the body has basically used up all of its ATP. So the muscles remain contracted. And that's why the body becomes stiff. All right. Now, after about 48, 72 hours or so, the proteolytic enzymes, the enzymes that break down some of the muscle tissue within the body and basically breaking apart the myosin head, those are going to start eating away at those, and then we're going to relax our muscles then. But again, we're going to be gone. We're probably not going to worry too much about it then. So that's your fun fact for the day. So a figure from your book that's basically describing what I just went through. Again, a current coming down the neuron, acetylcholine being released, going across the synapse, binding to a receptor in the plasma lemma, going down the T-tubule, stimulating the sarcoplasmic reticulum, releasing calcium, calcium binding to troponin, tropomyosin moving out of the way to reveal the binding site for actin, or for myosin, excuse me. And then we can have the muscular contraction then, because ATP is being broken up into ADP and PI, then it's being released, and then we can have the myosin pull the actin towards the center, and then we have muscular contraction. Then another ATP needs to bind before we can have muscular relaxation. Again, another figure showing exactly the same thing. All right, so let's switch gears here and go over muscle fiber types. So there's two basic fiber types, type 1, type 2. Type 2 is also broken up into two other subtypes, type 2A and type 2X. If you see a source, an article or a textbook that describes type 2B muscle fibers, they are type 2X fibers. Approximately 7-8 years ago or so, a lot of people kind of changed the nomenclature to from B to X. I don't know why they did it, but That's just what they did. So, but for our purposes, we're going to describe it as type 2X. So anyway, type 1 fibers, they have a larger mitochondria. The mitochondria are more abundant as well. And think about what's in the mitochondria. They have a lot of oxidative enzymes. So these type 1 muscle fibers are good for oxidative type of exercises. All right. So for like aerobic exercises, long runs, long cycling exercise, rowing, swimming. All right. Long distance events. All right. They don't fatigue nearly as quickly. They also have a lot of capillary density, which means they have a lot more blood flow going through them as well. And that's important because the more blood flowing through them, they're potentially going to have more oxygen flowing through them, which is good because they have more mitochondria and they need the oxygen basically to fuel the oxidative phosphorylation. Now, they tend to have smaller motor units, less than 300 fibers. Also, they're slow twitch. So the force velocity, they don't produce a lot of force very quickly. All right. And one of the reasons for that is that they have a slow myosin ATPase. So that ATPase that's bound to the myosin head is not as quickly hydrolyzed as what it would be on a type 2 muscle fiber. They also have a lower actin myosin ratio, which is again is not very good for producing high amounts of force. All right. Now, if we look at the type 2 muscle fibers, the motor units are really large. This actually should be, and I'm going to change this right now. That way you don't get too confused. There we go. You've seen the behind the scenes. So the motor unit is large, greater than 300 muscle fibers. These are fast twitch. They're going to produce a good amount of force really quickly. A lot faster than type 1 muscle fibers. Now, they have a fast myosin ATPase, which is one of the major reasons why it's a fast twitch fiber. So that ATP is split up a lot more rapidly than would be in type 1 muscle fibers. Now, they also have a highly developed sarcoplasmic reticulum, which means there's going to be a quicker release of calcium, which can possibly translate into faster binding to troponin and binding of myosin to actin and muscular contraction then. So, and coupled with the fast myosin ATPase. They also use ATP quickly, which means they fatigue a lot more quickly as well, a lot quicker than type 1 muscle fibers. All right, so they don't last nearly as long. And plus, they don't have nearly as much mitochondria. So therefore, they don't have a lot of oxidative enzymes. The capillary bed might not be as big as well. So those are the major differences between the two major muscle fiber types, as well as the sub fiber types. Now, the big difference between type 2A and type 2X fibers, type 2A fibers tend to have maybe a little bit more properties of type 1 fibers. They'll probably have a little bit more mitochondria than type 2X fibers, but they won't necessarily be totally aerobic in nature. They're going to be more anaerobic. All right, so looking at the three muscle fibers that we are describing, if we look at the maximal shortening velocity, so essentially the rate that it is shortening, you see type 1 muscle fibers aren't nearly as quick as type 2A, and type 2A aren't as quick as type 2X fibers. Now, the maximal specific force, basically the rate that the muscle is able to reach a certain amount of force production, type 1 muscle fibers really low. They're not able to to produce a lot of force very quickly. Now, type 2A and type 2X fibers are fairly close to each other, but type 2X is a little bit better at producing force really quickly. Now, power, type 1 muscle fibers don't produce a lot of power. Type 2A fibers produce a significantly more amount of power than type 1 fibers, but again, the type 2X fibers, they are basically the powerhouse muscle fibers. Right? They're going to produce a lot of force really quickly, and that's what they're designed to do. All right, the type 1 muscle fibers, all right, they're there. You're going to be using these throughout the day, anytime you go for a long run. Any type of aerobic exercise, you're going to be using mostly type 1 muscle fibers. Now, the type 2 A fibers will probably be using some as well. Type 2 X fibers, you're probably not going to use those a whole lot. Generally speaking, they're going to be fairly dormant throughout the day unless you're doing a lot of heavy lifting or just in your job or in your workplace, or actually doing strength training. So it needs to be a significant amount of force for those to be stimulated. All right, you can take a look at table 8.1, the characteristics of the muscle fiber types, and it describes fast fibers type 2X, type 2A, and then as well as like slow fibers type 1 fibers. Whew, that's a mouthful. So we've kind of gone in this in detail. I won't go over this too much, but you can take a look at that and get some information from that as well. Now, type 1 fibers during exercise, these are, again, going to be used for highly aerobic endurance exercises. All right, now, they don't need to be marathons, but anything greater than 3 or 4 minutes, you're going to start using more type 1 muscle fibers, or anything that's low intensity, really. You're going to be using more type 1 muscle fibers than anything else. So these are, again, for extended periods of time when oxygen is needed, because remember, they have a lot of mitochondria. So if you need a lot of oxygen for that exercise, you're going to be using type 1 muscle fibers. Now, the ATP is going to be coming from fatty acids as well as carbohydrates. At rest, it's probably going to be around 50-50. During low intensity exercise, you're going to be burning more fats than carbohydrates. During high intensity exercise, it's going to be more carbohydrates. So you're going to be getting closer to 100% carbohydrates. If you're at max exercise, you're at all carbohydrates. But if you're at max exercise, you're not going to be using as much type 1 muscle fibers as you would during a low intensity. So again, this is type 1 muscle fibers. They're going to be good for endurance athletes. Having a high number of type 1 muscle fibers is good for endurance athletes because, again, it's good for aerobic exercise. Now, the type 2 muscle fibers, on the other hand, these are going to be good for explosive type events, such as high jumping, long jumping, something that's going to be really quick. You need to produce a lot of force immediately, but they do fatigue quickly. All right. You can't continuously jump forcefully, your maximal height that you can jump repeatedly for hours on end. Those muscles, those type 2 muscle fibers that are required for that type of movement are going to fatigue really quickly. So again, the type 2A, they have more force, but they do fatigue a little bit quicker than type 1 fibers. The type 2A, usually short, high-intensity endurance exercises, maybe something like a 400, maybe 800-meter run, maybe even getting into a mile run. The type 2X fibers, you're really looking at short, explosive activities. I mean, again, this is stuff for you're jumping as quickly and as forcefully as you can, or you're lifting something that you need a lot of weight really quickly, or you need a lot of maximal force production. All right, the muscle fiber recruitment. All right, this is essentially looking at the order that the fibers are going to be recruited, and they're always going to go in this order, if you can see there on the bottom of the slide. So this is also known as motor unit recruitment. So less force production means that there's fewer, smaller motor units. The more force production means that there's larger or more motor units. All right, generally speaking, type 1 muscle fibers have fewer, smaller motor units, so they're going to produce less force. Type 2 fibers are going to produce more force, although they have more, larger motor units. So when you start to move your muscle, start to contract your muscle, start to pick up something, say something's on your desk, thinking if you're sitting at your desk right now, you probably have your book or something there. If you start to pick up your book, All right, you're always going to start off with using the type 1 muscle fibers. You're not going to start off with the type 2X fibers. All right, you're always going to start off with the type 1 fibers. When you're not able to overcome the force of that book, basically pulling down because of gravity, you're going to start using the type 2A fibers. And those are going to produce a little bit more force to try to lift that book. Now, generally in a textbook, you might not get to type 2A fibers. You might get to type 1. But if the book's heavy enough, let's say that book weighed 300 pounds and you're trying to lift it up. All right, you're still going to start off with type 1 muscle fibers. And then you're going to go to type 2 muscle fibers, and you might be able to start to budget a little bit right there. But to really lift it, you're going to have to use the type 2X fibers. So something like a 1 repetition maximum, you're going to need the type 2X fibers. So this is a figure. This one is not in your book, but it is important for kind of describing the motor unit recruitment. All right, so the smaller motor units, generally speaking, the type 1 muscle fibers, again, those are going to be recruited first. And if we look at this figure, we have 20 repetition maximum down here. So even down here, maybe you're looking at 30 repetition max and higher. All the way up at the top, you have 1 repetition max. And you have the activation threshold, basically the activity or the activation of those muscle fibers. So again, type 1 muscle fibers, they're going to be used for low repetition maximums or basically at the beginning of a lift or exercise. Now, if you need more force to either lift that weight or lift that object, then you're going to move up to maybe type 2A fibers, something maybe in here. So maybe these smaller blue spheres. All right. Now, if that isn't enough, then the activation of type 2X fibers is going to be needed. And that is way up here. We're talking maybe 2, 3, probably even just 1 RM. All right. That's when the type 2X fibers are going to be recruited. All right. It's always going to go in this order. If you lift a 5-pound dumbbell, it's going to go in this order. It's probably going to stop way down here. You're not going to need to activate the type 2X fibers. But if that dumbbell is 50 pounds, and that's all you can lift, that's your 1 repetition maximum for a bicep curl, you're still going to start off with type 1 muscle fibers, move up to type 2A, and then type 2X. All right, it's always going to go in that order. Having said that, you can train your body, all right, and this is a specific types of training over months and months and months. You can train your muscles to basically skip over the type 1 fibers and possibly the type 2A fibers. More than likely, you're going to skip over at least the type 1 muscle fibers. An example of this is a power lifter. A power lifter needs to generate as much force as possible very quickly. So they've actually trained their body. Good power lifters, they go through the proper training. They're going to train their body to basically skip over this because this is kind of wasted energy, wasted movement down here. And they're going to skip over these and they're going to start with either type 1 or type 2A or go straight to type 2X. Another example of that would be a pitcher in baseball. They don't need type 1 muscle fibers really all that much as soon as they get through their windup and they're starting to go towards home plate. They don't need the type 1 muscle fibers in their arm to throw quickly. They need the type 2A fibers or the type 2X fibers. So over those months and even years of training to pitch properly, proper form, they're going to train those specific muscles to skip over the type 1 muscle fibers and go straight to the type 2A fibers when they're wanting to produce as much speed in their arm, thus translating into more speed to the ball, to home plate. All right, so looking at the length-tension relationship, essentially what this is describing is that the muscle is weakest at a basically either fully contracted and fully extended. All right, somewhere in the middle is where it's going to have the most muscular, or at least potential muscular force. All right, so what this means is if we look at this condition right here, this is actually this one right here. And the one in the middle is the 225. And the one over here, this is fully extended or fully stretched out. This is 365. All right. No, don't really need to know about the units or anything, but basically what's happening is this is in a fully extended muscle. Say you're in a doing a bicep curl. This is when your arm is completely extended, and then about halfway through, maybe a little bit less through that movement, this is where the muscle fibers are. All right, so we have myosin and actin. All right, so the actin isn't quite near the M line yet. Now, when we have full contraction, we actually end up either having actin together or there could be a slight overlap. Now, again, you're not able to produce as much force when it's fully contracted, and you're not able to produce as much force when it's fully extended or fully stretched out. So a slightly stretched muscle in this condition here, slightly stretched compared to fully contracted, is going to be much more efficient at producing a large muscular force. And it has to do with the cross bridging. So there's a maximal amount of cross bridging and maximal tension development that is occurring in that situation. Basically, when you go through an exercise, is that you'll end up coming across a situation somewhere where you're going to be strongest in that certain exercise. And in a bicep curl, it's going to be at a certain angle that your elbow is at. All right. And basically that biomechanical efficiency or that maximal amount of force going to be produced is going to be dictated by this right here. All right. That maximal amount of cross bridging that can occur. All right. So again, fully contracted, not as much cross bridging. Fully extended or fully stretched out is also not going to produce nearly as much force. Now, this fully stretched, and why I say stretch is because one of the theories behind the reason why static stretching, just static stretching, okay, I'm not talking about dynamic type of stretching. You can look those terms up, but true like static stretching where maybe you just warm up for a little bit, maybe go for a short run, and then all you do is just stretch. All right, you're not doing really any activity while you're stretching. That is not really the best thing to do. In fact, for athletic purposes, it should be avoided. You should be doing something else. It should be in combination with certain movements. And again, look up dynamic type of stretching. It's going to be more beneficial if you're a coach. If you're in athletics, trust me, the dynamic stretch generally is better than just static stretching by itself. All right. Now, static stretching could possibly stretch out the muscles, maybe similar to what this looks like right here. Thus, you're not able to produce nearly as much muscular force. And so if it's in a sport, maybe like weightlifting, that's obviously going to be bad. I mean, you want to produce as much force as possible. So you don't want the muscles to be completely stretched out because you're not going to be able to produce that force. But in really any sport, if you think about it, the more you stretch this, the weaker the muscles get, basically. So say if it's a sport like tennis and someone is cutting, all right, they're ready to hit the ball and they step real hard. The muscles around the knee, if all they did was static stretch, all right, and most static stretches, if you think about it, especially in the legs, are just stretching out the hamstrings and muscles around the knee. If you stretch those muscles out too much, that could weaken them. Therefore, the knee isn't going to be as strong, so you could actually cause an injury from static stretching by itself. So some things to think about. Again, those are some things you really ought to look up. There's a lot of research on it. Trust me, the static stretching by itself isn't as beneficial as what we once thought it was. All right. So anyway, I'll leave it. I'll leave that there, and you can go look up some other stuff on static stretching. But that's kind of your extra information for the day. All right. So looking at force velocity. So as the velocity of the movement increases, the force that the muscle is able to concentrically generate decreases. So there's a negative relationship. All right. Concentric contraction is the muscle shortening. All right. There's going to be tension within the muscle, and at the same time, the muscle is shortening. All right. So as the velocity of the movement increases, the force of the muscle to contract in a concentric motion. All right. So if you look at a bicep curl, this is going to be going up. All right. Shortening the bicep. So it's determined and limited by the maximal rate of the myosin and actin that's able to attach and detach to each other. All right, so there's a little bit of time that needs to take place between myosin attaching, moving actin towards the M line a little bit, and then it's going to detach and attach to another binding site. And then it's going to keep on doing that basically until actin reaches the M line. All right, so that takes a little bit of time. So you're not able to produce as much force as you would be during maybe eccentric contraction, at least during the same amount of time. And we'll see that here in a second. Oop. So an example of this would be if you were to go on a bench press and you try to do a 1 repetition maximum, you're going to lift that weight really slowly. You can't lift it extremely quick. All right, if you can lift it extremely quick, that's probably not your 1 RM. Now, if you take half of that weight, all right, so say your max bench press was 300 pounds and you got put 150 on the bar, you're going to be able to lift that a lot quicker than you would that 1 RM. All right, now going up pushing the bar up is a concentric movement on the bench press. So again, as the force increases, the velocity is going to decrease. As the force decreases, the velocity increases. So again, there's still a negative relationship there. Now, on the other side of that, if you look at the eccentric contraction, all right, a lot of times it's described as extension. All right, there are certain circumstances where that's not really the case. Eccentric contraction, the definition of that is there is tension, there is cross-bridging within the muscle. All right, so there is a muscular contraction, but at the same time, the muscle was lengthening. So in an eccentric contraction, there's a positive relationship. So as there's movement, as the movement increases, the force will also increase. Now, most of this is thought to be caused by the elastic properties of the muscle, basically the stretching of some of the muscle tissue, some of the ligaments. It's able to resist some of that force a lot quicker, and you're able to produce some of the force much quicker to overcome that force quickly. All right, and also the eccentric contraction is also important for muscular damage. This is where most of the damage is going to occur in those Z discs. All right, and that's going to occur, generally speaking, within the eccentric contraction. All right, because you're trying to lengthen that muscle. You're basically trying to pull actin and myosin apart, all right, the opposite way, but you're also, at the same time, you have contraction. So you're trying to move them towards the center at the same time. So there's a lot of basically opposing forces going on there. So it creates some overstress within the muscle, and that weak point most of the time is going to end up being the Z discs. All right, that was chapter 8, skeletal muscle, the structure and function of skeletal muscle. So again, if you have any questions, don't hesitate to put a comment below, as well as email me if you need to. Thank you for watching. If you have any questions, any comments, let me know.